this pain, I can never get away. Dealing with the bullshit every other day. Close my eyes and I can feel the hate. Close my eyes and I can feel your pain. Searching for the good things in life. Recently I can't find that stride. Broken hearted with a bloody knife. How you end up most of nights. Paper hearts cause of broken thoughts. Life is slowly falling apart. Try to fix it but where to start. Try to fix it but where to start. 5 steps forward, 10 steps back. Life refused to give you some slack. Be the big guy and don't look back. Save your heart some heart attacks. Pushed away all my fucking life. I ain't never seen the fucking light, but I'm still here till I fucking die. And there's still tears every single night. Try to work on myself and me. Fucking up but I still believe. Head is high even though I'm weak. Won't live on my fucking knees. And that's a promise that I try to fucking keep. Try to keep. 5 steps forward, 10 steps back. Life refused to give you some slack. Paper hearts cause of broken thoughts. Life is slowly falling apart. 5 steps forward, 10 steps back. Life refused to give you some slack. Paper hearts cause of broken thoughts. Life is slowly falling apart.
Good afternoon. Okay, good afternoon all, and thanks for coming to the lunchtime session. I was going to kick off a few announcements just before we start. So just a couple of talks where there's a few changes. So the talk by Mariska, she's unfortunately not here, but a Jan Bester will provide us with a quick talk about an initiative with regards to forest ecosystems. And then Stephen Hoodborn, I'm also just giving you a round for the whole session. Stephen Hoodborn's unfortunately also not here. Albert Myberg will be giving his talk. And then Stephen Koza's also not available. And the talk will be done by Peter Botha. Peter, are you handing off? Okay, thanks, man. Okay, so off we go. And our first talk is Dr. Heidi Hawkins. Thanks, Heidi. You can go ahead.
So I'm going to be speaking about soil organic carbon predictions from biogeochemical models in both wildlife and livestock systems, and just questioning, how certain are they? But hopefully it's not just a cautionary tale. I'll also make some suggestions about what we can do about that. And I'd like to acknowledge contributions from many students and also colleagues around data and discussions. So most carbon is below our feet, unless you're swimming in the ocean where there's lots of carbonates. But we're going to be talking about soil organic carbon, and most of that is in the grasslands and the savanna if you're in South Africa. So forests win on soil organic carbon, or SOC. If you're looking at concentration, that's the darker areas on this SOC map. But grasslands win on SOC amount, and that's because of their extent. And so grasslands would then be followed by savanna and Namakuru and Fynbos. So there's a challenge in managing open ecosystems when they may be more or less altered. So we all know that natural disturbance like mammalian herbivores and fire drive heterogeneity, and this is part of ecosystem function, and soil organic carbon storage is part of that. But there's been simplification of herbivore feeding communities, along with sometimes reduced fire, so sort of a prevalence maybe of grazers and less mixed feeders. And we know that's linked to increased woodiness. And in some cases, also to decreased SOC. Example is a paper from the Klee experiment, Sitters et al. So now there's interest in not only around forests, but also open ecosystems to manage them according to so-called natural climate solutions or nature-based solutions. And one popular vehicle for that is carbon finance or credits. So if you're a reputable carbon standard, then you require forecasts from biogeochemical models, and these must be peer-reviewed, but they can vary widely in their complexity. So just for example, the daily century model is a complex model. It requires over 20 inputs, and you can fiddle around with all sorts of plant parameters like carbon to nitrogen ratios and initial soil values. Daily climate is an input, soil properties and land use. And then you get lots of outputs, but mainly it's about trace gas fluxes between the atmosphere, the soil, and plants. And you get plant production as well, and you also get sequestration into various soil organic carbon pools. Now I'll come back to this, but these pools are often called active, slow, and passive, and this supposed to relate to their stability in the soil or their residence times. But these are actually completely hypothetical. But anyway, my point is then you also get very simple models like this SNAP model based on the Serengeti National Park. It only has 5 inputs and depends heavily on those in bold, and you only get one output, which is SOC at equilibrium, and it's also not clear how long that equilibrium takes. So whichever model you use, it must be validated, and then of course you can create scenarios for your carbon project around changed management or desired, whatever your desired management is with fire and herbivory, and then also look at future climates. So I'm going to show some examples. This is high altitude grassland and wildlife system. We adjusted descent using the Brotherton trial, which was established in 1980. You can see it here. We sampled outside the burning trial to get herbivory and annual fire, and then we sampled in the park to get herbivory and biennial fire in the Drakensberg. And then inside the trial we could get annual fire, biennial, and then no disturbance. And so we get some reasonable R-square between measured and modeled herbaceous biomass with, the highest amount of biomass, as you might expect, with lower disturbance. I won't explain all the colors, I'll just point to them. This is no disturbance, and then that is biennial fire, and that's herbivory and biennial fire. And then this is the largest amount of disturbance here with annual fire and annual fire plus the herbivores. So you can see that, especially for some of the treatments, the model overestimates herbaceous biomass at low biomass levels. We also get a reasonable overlap in measured and modeled phenology. So the dark, this is just the biomass over time, and modeled is dark, and the measured is the dark blue, and the modeled is the light. And we get also an overlap in SOC between the measured, which is blue, dark blue, and that lighter blue, which is the model. So that's what you want to find. You really want to get good overlap in biomass and also in the thing that you're interested in. So in our case, it's SOC. These models also measure water flows and other gas fluxes, and if you're interested in those, then you have to measure those. So we get the same sort of trend here, basically higher SOC in this long-term trial where you've got biennial fire or moderate fire with some herbivory, or just biennial fire. So moderate disturbance. Okay, so we could have done all sorts of scenarios for Brotherton, but we actually took this to a grassland livestock system. This is the Imvenyani communal area near Kokstad. And that community agreed in 2016 to have planned grazing, where it's a really simple sort of 2-camp approach. One area is rested and the other's grazed as part of conservation agreements. And again, we just have some overlap between measured and modeled herbaceous biomass, only in the unrested, which is that one here, because in fact the rested areas were not consistently rested by the community. Nevertheless, we can use these models as a heuristic, and actually last week we were there showing them or discussing these data with them, looking at various climate scenarios. I'm just showing RCP 2.6 here, but this is the next 20 years, and we could see that, okay, there's more biomass generally with a triennial fire. And then again, don't worry about the colors. I'm just going to point out what we could say to them is that, compared to high stocking rates and continuous grazing, one could increase herbaceous biomass by having recommended stocking rate and the simple rotation. And just focusing on the triennial fire again, very much the same trends for SOC. But the point here for this talk is that the emissions removals that are predicted by this model are really quite high, around 6 tons CO2 equivalents per hectare per year for 20 years until you get a saturation. I'll give you one more example. in the Savanna, also a livestock example in the Kruger Canyons biosphere. So here are some communal grazing areas that we sampled next to various nature reserves, and there's Kruger. We also took it all the way up to the Blader River Canyon area and grasslands. And we applied this simpler SNAP model, and we did get some overlap with of the model with the measured, but it's not performing very well at the moment. You still have to work on it or ditch it. Anyway, we came up with some scenarios, and with reduced grazing intensity and moderate fire, you are predicted to have higher SOC. And so this is actually a carbon project that has reached past the feasibility phase, and the emission removals are predicted to be between 1 and 3 CO2 equivalents per hectare per year, which does qualify for a carbon project. It would, in theory, then be financially sustainable by FY26 from the sale of carbon credits that would go to the community. However, at the moment, this really needs to be backed up with day-cent at least, but also ideally something more sophisticated based on the latest knowledge around soil organic carbon. So we've got these predictions from models, but if we look at the SOC response to disturbance, it varies a lot in reality. So here we've got fire, herbivory, this is wild herbivory, and fire plus herbivory in various long-term trials. So there's that high-altitude grassland Brotherton, lower altitude Ukalinga, and then the Nkuhlu and the Lataba trials in the Savanna. And what we have here is the least squared estimate in a linear model, where we're comparing these to the full exclosure situation where there's no disturbance. So 0 would be a neutral effect, and then you've got negative and positive. And so what we see is for fire, for example, we have Ukalinga with a negative effect, but Lataba and Brotherton with a positive effect. And then for herbivory, or the combination of fire and herbivory, we only saw negative effects. So I'm just showing the significant effects here. And then looking a little further afield, there are other examples of no SOC change. Obviously, this is not an exhaustive meta-analysis, but for example, in grazing exclosures in shrublands of Mallorca, we see no change, despite that drastic above-ground increase that you see in exclosures. You don't see it below ground. And similarly for semi-arid savanna in Ethiopia and savanna woodlands in Burkina Faso. So what are the possible reasons for this disparity between the very consistent projections from the model and what we see in reality? Well, many models may overestimate SOC, and that's because the SOC in models is actually quite generally poorly constrained in these process-based models. We may be underestimating the time because carbon fluxes are really quite small compared to the large mass of soil, and so it may take 50 years or more for us to see changes, and often what we're measuring is some legacy of the past. And then most models deal with imaginary SOC pools. I mentioned the active, slow, and passive, and they're unrelated to measurable fractions. And then other issues, we may be inadequately sampling the soil heterogeneity by taking point samples just in the open or just under canopies, and we may be missing the entirety of what's going on. And then inadequate analysis, maybe we shouldn't be looking at different fractions instead of total SOC. And then finally, context may drive SOC more than any sort of disturbance. So those are lots of areas that we can investigate, but meanwhile, what do we do around this uncertainty? Well, obviously we can try to use models like this one here, which does make use of measurable fractions. We can try and measure heterogeneity, But what we can do immediately is avoid tunnel vision around carbon in these projects. And we should measure and value other aspects of of soils, such as water holding capacity and fertility, and also the co-benefits, so-called co-benefits in these carbon projects. So we can, such as biodiversity, water, and benefits that may come to people with some desired change in management. So these carbon projects are abounding, and we can either make use of them or not. So thanks very much to the funders and to you for listening.
Thanks for the great talk, Heidi. We can squeeze in a question, one question maybe. Does anybody have a question? Right at the back there, thanks. Okay, it's fine.
Well, I can use the microphone. Heidi, if you can just clarify, it looks like when you mentioned kind of which land uses would result in potential change in the moist grasslands. You mentioned fire there. You didn't mention kind of fire and herbivory because obviously a lot of your potential client uses both fire and herbivory when it comes to kind of livestock farms.
Yes, so there was the combination. So in the different long-term trials that we measured, there were combinations of, just herbivory, just fire, or the combination of the two. And then in the communal areas, the one that I showed, they tend not to have planned burning. It may just happen any time. And so there we relied more on MODIS data to see what happened in that area, and then played around with different fire scenarios in the predictions. I'm not sure if that answers your question. Well, yeah, well, it's not sort of good or bad. It's sort of, I suppose, whatever the desired management outcome is. But to your point, I think that the reason there's so much variability in findings is that we're looking at examples with different fire intensity. They may have the same fire frequency, but different intensities. And the same with herbivory. Unless you're really defining those, it's hard to compare them.
Thanks.
So at least if you can verify your model based on something that's really known, like in these trials, we know exactly what the grazing frequency or amount is, and same with the fire, and then you can set your model with that, and then the model's useful as a heuristic to play with, any number of fire frequencies and intensities, but there's limitations in these models in how they define fire intensity. It is based on data, so they look at, the heat of the fire and all that sort of thing.
Okay, thanks, Heidi.
Yeah.
Thanks. That's okay. You guys can pick it up during tea or lunchtime. I'm sorry to break you, just to keep on schedule. But thanks for your talk, Heidi. Okay, so just quickly, just another announcement for the guys that are very interested in joining the think tank on nature-based solutions. There'll be a register at Jackie at reception. You can put your name on and your email address that the guys can send you an email with all the information, times, and a map to get to the SSRI conference center. But thanks, Corley. So up next is Corley Kutsia, and she'll be talking along the long-term changes in hoodie density in an African savanna. consequences for soil carbon. Thanks, Corley.
Thank you, Robin. Thank you for everyone that's here listening at my talk. This is quite a substantial body of work, and there's been many co-workers, and some of them are here. And I have to thank all of them, because without them, this work wouldn't be possible. And then one very important partner is Sapienza, the University of Rome, that has sponsored a lot of getting the long-term aerial photos georeference, which was a very lengthy and costly process. So this is a photo, aerial photo taken in 2018. You can see Pretorius Kop here, the camp at the bottom, and then one of the experimental burn plots just there at the top. This is Kambeni. And it's a little bit hard to see in this photo, but it's actually quite thick. The bush here is rather thick, and if you use your imagination a little bit, we had to turn a color photo into a black and white photo, because we're trying to compare it with these photos taken in the 1940s. If you go on the ground and you have a drive through Pretorius Kop, this is what the burn plots look like. This is just not photos from Kambeni, it's photos of Shabeni, which is another one of the strings. And it can get quite thick. So this is these are the plot at Shabeni that never burns, and then this one is the one that burns every year. So even though it burns every year, it is quite open and grassy, but you have like quite a lot of shrubbiness also in these plots. And then Lawrence very kindly went driving through Pretorius Kop a few days ago, and he took some photos. So this is just kind of the general area. Once again, there's plenty of open area, but if I drive through Pretorius Kop, I notice that it's rather quite thick, and a lot of it is terminalia. So if you look at this area in the 40s, this photo is from 1944. You have the camp again at the bottom. This is the area that then became the Kambeni experimental burn plots. And it's a very open area. And if you don't believe me, I managed to track down some photos taken from the 1920s. There's definitely some trees around, but they used to be quite large herds, and you have this rather open landscape. I think that is the Pretorius Kop, which you don't see from the camp anymore. And the description here is an open parkland landscape with sparse woody vegetation and numerous wildebeest and zebra. And I found another one of the area. It was taken in black and white and then color in autochrome, so it looks almost like painting. But once again, a very open landscape. So it's interesting. It's interesting to me that it appears to be thickening up in that area. But if you look at the long-term fire frequency, then the kind of redder colors here, and that is Shabeni there at the top, and this is Kambeni. They are situated in areas that burn frequently. This is, thanks Sinead did this, or I used a map that Sinead made. It's the number of times that the area burned out of 40 odd years, and you can see that these red areas burn almost annually. So what we then did is we just went and we looked at what happened on the plot in this time. And this work was done by Linda. Thank you very much, Linda, for sending these to me. Linda's been an absolute star in trying to help us. So this is the area that would become, in future, the no-burn plot at Shabeni. The same plot in 1965. I don't know if you guys can immediately see some manager obviously got it wrong and burned the no-burn in 1965. And this is 2012 and 2018. We started using 2018 photos, but that was just after the drought. So they were actually a little bit very dry. So then we kind of decided to compare the 1940s to 2012. Linda then just went and looked at, kind of enumerated the woodies in these photos, and it went from about around 20% cover to 35% cover. And then this is the extreme on the other end, the annual burn, 1965. The annual burn didn't burn at that point in time, 2012 and 2018. And if we look at the woody cover, it stayed more or less the same, 23%, 25%. And this classification just picked up the surroundings were actually burned. So we had these 4 questions. How did woody cover change across the EBPs in the 70-year period? Can we find a correlation between the woody cover as it is now or the woody cover change in soil carbon? And if we find more soil carbon, is this a tree effect? Where's the soil carbon coming from? And then we wanted to see if we can scale all of this up to this kind of sandy soils of southern Kruger. And we have looked at the woody cover remotely from the aerial photos, and so both Linda and Tulia, that was going to talk on Thursday, is working on this using different methods. I also looked at actual surveys that took place on the plots, and to kind of use allometric relationships to work out biomass. And then we went and we measured under canopies and away from canopies soil carbon at 4 different depths. Okay, so looking at this first question, these are just 3 plots. So we have woody cover. This is the biomass that I got from using those allometric relationships. The woody cover is worked out from the aerial photos, and then that's just a change over that time period. And as you can see, like, woody cover was quite low. This is the Tuescorp and Sakusa treatments together of the annual, triennial August, triennial Feb, and the no-burn. Okay, and it's the same sequence throughout. So cover was quite low across the EBPs in the 1940s. And then the treatments had an effect. Even in the annual burn, we are gaining woody cover, and then as we expect, like largest woody cover in the no burns. The biomass patterns were not that clear. I mean, there's problems of allometric relationships, and they use different survey methods in that period, so the data is a little bit wonky. And you know, those guys, I thought they had so much time and resources and money back in the 50s, but they left out the Feb 3 treatment. They didn't enumerate it. Such laziness. And yeah, and then if you look at woody cover change, you can see like huge increases, mostly on the most severely on the no-burns. Okay, so is there a correlation between woody cover and cover change, or cover change in soil carbon? And we found that a model that looked at woody cover change and the clay to sand ratio best explain soil carbon. The correlation with woody cover per se was not so strong, but I'll get to it. And then there was also a correlation between woody cover change and the fire frequency. So we just went, with the help of Tesha, we counted for this whole 60-odd years of fire experiment how many times each plot has burned, and then we just worked it back to a ratio out of 1. So we just didn't include this in the model because there was that correlation between those 2 variables. And if I kind of sum it into a 3D little figure, that's what it looked like. And then lastly, we also looked at, and let me just quickly explain what we did, is we did soil carbon at a plot level. So we knew how much carbon there is on average under canopy and away from a canopy, and then we corrected for the woody cover on the plot, so that we kind of get an idea of what the carbon would be per hectare. And So the last thing we did, and that was for all 4 our depths to 30 centimeters. We also just went and looked at our very shallow depth, which was 0 to 5 centimeters, and then we had it as a ratio over the deep carbon. So as we see woody cover increase, that carbon that you were gaining in the topsoil also increased. So where did this carbon come from? Is it from trees? So this is just a plot where I looked at soil carbon and then looked at which, so we used carbon isotopes to see which carbon comes from woodies and forbs and what amount of carbon comes from grass. So you do get a slight increase of tree-derived carbon with an increase in woody cover, and there's kind of a non-significant decrease in the grass carbon. But you can see for most of this woody cover range, grass carbon is actually winning out over tree carbon. And then this looks like a little bit of a rendogram, but this is soil under grass, soil under trees or under canopy, and then the total soil carbon. The light green is the carbon that we get that is grass derived, and the blue one is the carbon that is derived from trees. So what you see here, you have, as expected, a lot of grass carbon under grass. But then I was very surprised to see that on average you have more grass carbon under trees. Then if you sum them, grass carbon is actually winning out. It's only a year after not burning for 60 years that you have your tree carbon starting to approach grass carbon. And I thought that that was a very interesting result. I hope everyone is as excited about it as I am, although some of our previous work has found the same in different systems. Okay, and then the million-dollar question, and this is the million-dollar question, right? What is the implication now? Can we now say that Southern Kruger has been gaining woody cover, so yay, we can sell our carbon? And I'm not going to answer that question because Tulia is going to answer that question on Thursday. So you guys are going to just have to stick around until Thursday at 11 o'clock, and then Tulia is going to tell you whether we're going to be making big money or not. So just to quickly sum it up, yes, we have been gaining woody cover over the southern EBPs, even in places where we burn annually. Is there a correlation between woody cover or woody cover change? There isn't a clear correlation. I was envisioning this work to just give me a really nice correlation between woody cover and carbon, and that would have been so awesome and wonderful, but it didn't work out that way, as ecology often doesn't. But at least there's quite a clear correlation between woody cover change and soil carbon. And then lastly, where is this carbon coming from? And this carbon is coming, yes, a little bit is coming from trees, but you have a lot of grass carbon in any case. So having extra trees in your system is just allowing it to gain a little bit extra. And I don't think it's really worthwhile suppressing fire for 60 years to gain that tiny little bit of extra carbon. So that is my carbon story. I just have to say, also beyond all those co-workers, there's been a whole team here in Kruger, technicians and field research assistants that's helped us with gathering the soils. Soil sampling was quite a large effort. So thank you for everyone in scientific services.
Thank you, Corley. That was a lekker talk for after lunch. Really interesting, thank you. We have time for 1 or 2 questions. Okay, I think was it Susan with the purple on? Yeah, Susan and Heidi. Is that okay? Susan, you can go ahead, and then Heidi can go.
Yeah, great talk, Corley, thank you. I'm just trying to imagine what would happen if you let this continue and you'd get the extra trees and you'd get the extra tree carbon. what would happen to the grass carbon? I mean, it would obviously stick around for a long time, but, you know, are there sites where encroachment of what was a savanna has been around for so long that you could look at the effect of that? Would the increase in tree carbon in the long term make up for the ultimate loss of the grass carbon being replenished by grasses?
Yes, so that's a bit of a caveat in Kruger is you have elephants, and even the most change that we have is about 40–50%. So we're not approaching 80% and losing grasses completely. So I think that's a very good question. When you starting to lose your grass carbon, will your tree carbon be able to compensate? And you know, this is just, I think Louis would be a good place to answer that question. Yeah.
Oh, thanks. That's just so interesting because reassuring in a way, because I've just been measuring soil carbon under wattle and just assumed that it would be so much higher than the grass next door, but it wasn't. And that's after 30 years of invasion. So I don't know what's going on, but it's interesting.
Oh, that's also nice for me to hear, Heidi. Because, yeah, I was just thinking, okay, we're missing out on the kind of thicker side. So, you know, you might start seeing different patterns. Thanks for the comment.
Okay, thanks, Collie. Well done. Okay, so we're running slightly ahead of schedule. So Jan Bester asked if we could move his little 5-minute talk to the end of the session, which I said it's okay. But sorry, Isabel, I think if Isabel, are you around? Isabel, hi Isabel, sorry. So thanks Isabel, if you can come up. Isabel will do a quick speed presentation on below ground water use traits of savanna trees and grasses.
Hello, everyone. Sorry. So a bit of background. Many of the models that seek to explain tree grass codominance focus on this idea of niche partitioning and the trade-offs related to water use and acquisition. And this is because we know things like rainfall regimes can influence tree covers and savannas, and thus their tree grass ratios. Despite this importance of water to savanna plants, there's not that much research comparing the roots of trees and grasses in savannas. An exception to this is rooting depth, which was widely investigated as an early theory of tree grass codominance. But it was built on these assumptions that grasses have a greater ability to transport water and exploit soil moisture, but we lack the data to back up those assumptions or quantify other key root functional traits, such as conductivity. So we conducted a greenhouse study with around 40 species of trees and grasses native to the savanna. And those are the buckets we grew them in. So a little bit about our methods. We first looked at the vascular anatomy of these trees and grasses. And so you can see here we had over 500 cross sections of root slides like this one, and we isolated the xylem area. So it's important to note in roots, you can broadly split conductance into two groups, axial transport and radial transport. Radial transport refers to the movement of water into roots, whereas axial transport refers to the movement of water through xylem vessels. So since we only quantified xylem area, we were then able to determine the theoretical axial conductivity of our roots based off of these root cross sections. For the same plants we had all these root cross sections from, we also have their relative growth rates. And then finally, for a cohort of plants using just trees because of limitations of our apparatus, we quantified the whole root conductance value for an entire root system of a tree. And then here you can see some of the really cool cross-section slides of trees and grasses, and you can kind of start to see the differences that poke out. But just a note on our whole root conductivity, what we found was our values of theoretical axial conductance or conductivity were related to whole root conductance in most of our tree species, and it was significant. Or there's a variety of root morphology within our species, with species like marula up there in the right-hand corner having extremely high conductivity. So what we found from the analysis of vascular anatomy is clear differences in terms of the vessel architecture of trees and grasses. Grasses had larger vessels, greater lumen fraction, and greater theoretical axial conductivity. And trees really just had greater number of vessels. And again, all of these differences were significant. And then when we related these vascular traits, here I show theoretical axial conductivity to growth. We found that in both trees and grasses, these vessel architecture traits are related to growth, indicating that vessel architecture is an important component of tree and grass growth in savannas, and that water may be an important component as well. So in conclusion, we found support for our values of theoretical axial conductivity as an important component of whole root conductance. There are clear differences in the vessel architecture of savannas, trees, and grasses, and these differences are linked to growth rate. And overall, this supports the idea that trees and grasses have different niches in terms of their below-ground water use and transport, and backs up the assumptions that the rooting depth initial hypothesis was based off of. Thank you.
Thanks, Isabel. We have 1 minute for one question. Does anybody have a question for Isabel? No. Thanks, Isabel. Next up is Jessie Neppard, and Jessie will be presenting on the root form and function in grassy ecosystems. Thanks, Jessie.
Thanks for having me here today. I'm going to give you a quick synthesis talk over some projects that we've been working on lately. This data comes from some of our North American experiments as well as some of the work we've been doing here in South Africa over the last 10 years or so. I do want to acknowledge my collaborators. I want to pay particular attention to Rachel and Emily, two of my younger collaborators who helped me collect the data and then went through countless versions of this talk to give critical feedback, as well as my older collaborators, Ricardo and Tony, who didn't help collect any of the data and were unaware I was even speaking. And so, when we think about the role of roots in ecosystems, there's a lot of different questions that we can start to think about. The below-ground processes and grasslands of savanna play this unique role in this ecosystem, more so than almost every other biome on Earth, in terms of thinking about the roles that roots play for cycling nitrogen, carbon, water, impacts on above-ground processes, etc. And for this reason, there's been a considerable amount of work that has been done to try and understand these processes. But working below ground is significantly difficult. It's hard to excavate, it's time-consuming, you break the roots, it's just very laborious. In addition, what happens above ground tends to translate below ground. We know that both bottom up and top-down drivers play significant roles on root communities below ground for both woody and grassy systems. As Isabel just noted in her talk, when we think about ways to quantify roots, for over 100 years now, we've been looking at traits that get at like the maximum rooting depth of roots, as well as snapshots of biomass, typically in the top like 10 to 20 centimeters. But we know that the zones that exist in the soil not only have differential amounts of biomass, root length changes, and it forms basically ability to utilize water from discrete zones. Just because roots are present in one zone of the soil doesn't mean that they're active in that zone of the soil. So we need to start moving away from this idea that maximum rooting depth means anything, because it typically does not, unless you're using a very coarse global model, and start to focus in on some of these other questions. So how do we utilize these other characteristics besides maximum rooting depth for greater clarity in our grassy ecosystems? One thing that we've been working on is trying to find ways to quantify below ground net primary productivity beyond just that surface soil layers. So we've been using these steel cages that go a meter deep that we can install in the soil. We pick all of the roots out of that soil, put it back in the way it came, then we can extract it, and then we have these meter-long cores over here to pull all the roots out to quantify below-ground net primary productivity. This provides very detailed estimates of what's happening below ground in terms of root growth. And when you start to compare grassy ecosystems versus woody, grassy portions and woody portions of the same ecosystem, you can start to get at changes in root length as a function of fire frequency. You can get changes in biomass, by depth. And you can get changes in specific root length, which is one of the more common traits that is utilized in these systems. And just as a reminder, most studies that do this only utilize that top 20 centimeters of the soil profile at maximum. So when you start to get deeper into the soil profile, you can start to get at dynamics that reflect what's happening below ground, and then how that information is being translated to root production, which then, of course, impacts carbon storage significantly into the future. So this is for BNPP. You can also compare this to just standing biomass. These are from the same locations. One of the things that's interesting to notice for both the grassy and the woody systems, even though these are perennial plants in a very high growth system, a large fraction of this biomass and root length is turning over on an annual basis. And so that's quite interesting to me that when you think about these roots that are less than 1 millimeter in size, even though these are perennial systems, there's a high amount of turnover that's taking place. We tend to just think about these shallow fine roots, but root distributions we know are not uniform. And so when you want to look at the coarse woody roots that exist in the system, like think about what Corley was just showing us a moment ago, if you want to sample for that, a snapshot location that you choose to sample for coarse woody roots might miss most of those coarse woody roots, especially when your core is only a few centimeters in diameter. So one of the things that we've been doing to get at this is excavating entire below ground assemblages of clonal plants that are invading our grasslands. These are our woody encroachers. So this is 1.5 meters by 1.5 meters by 1.5 meters. You can see my students down here below the root system as well as above it. We've been excavating these whole islands to create three-dimensional models of these woody coarse root systems in situ, to give us more information in terms of how these roots are being allocated at different soil depths, how big they are, and then what the implications might be. One thing of note, this data hasn't been worked up yet. This is the student in the pit down here. This is part of his master's. But one thing of note is the above ground stems here of this clonal plant that has rhizomes going on either side of the pit showed no signs of leaf stress, despite the fact it had no soil on its roots, and it was 35°C for the week that it was sitting out there being exposed. So some of these encroaching woody species can move substantial amounts of water through their lateral rhizomes, basically to keep their veg above ground hydrated despite extreme stress. Like not having any soil is a pretty extreme stress. So that's part of the carbon story of our root work. Moving on to some of the water, how can we think about linking root abundance to functional water uptake? How do these root traits provide insights into adaptive responses to availability? And then the last one, how do we scale this up across the landscape? So typically when any of you who've seen me talk before, I'm an isotope person. Almost all of this type of work that I've done looking at functional water uptake is using stable isotopes. There's been tremendous amount of fantastic work on this topic here in Kruger, whether it was Andrew Kolmatiski 10 years ago, or whether it was Maddie Case and Carla Staver last year. There's been so much fantastic work on this subject here in the park. One of the ones, though, that I want to point out that's a different approach that has been grossly underutilized is a technique pioneered by Ricardo a few years ago, where he looked for correlations between sap flux and then statistical relationships with changes in soil moisture at different depths. And Ricardo's approach clearly showed that for Terminalia and Pterocarpus, soil moisture increases at deeper soil depths were strongly positive with terms of an increased sap flux, meaning more water movement. By comparison with grass species, you see an opposite relationship, where that correlation and changes in surface soil moisture. So it's getting at this paradigm that Maddie's work and Andrew's work and a whole host of other people have shown that there's this very distinct niche partitioning happening in this system. Isabel just talked about the value of root anatomical measurements, and I will also beat on that drum. This is another grossly underutilized technique that with a patient student and some microscopy time, you can develop an insane amount of information. So one of the things that we've been proposing and been doing for a while in our lab is using root anatomical measurements to infer physiological functions. Some of the things we can do are look at total conduit area, diameter, and number, You know, similar to what Isabel just showed you, the steel area, which is there in yellow, the thickness of the endoderm, the thickness and area of the cortex, and then this thing here at the bottom called conduit wall thickness, and it's expressed as this T over B. So you can think about the thickness of this wall here on the conduit divided by the diameter. And higher values of this tend to equate to species that have more safety against water stress. So when you look at this, this is grasses versus woody plants. As you go down in the soil, so it's the same root, but then you're measuring these characteristics at different depths of the soil based on different burn frequencies. So 1 and 4 year burn for the grasses and a 4-year and 20-year burn for the woody plants. And you can see differential relationships between the woody versus grassy plants despite being measured in the same locations. But one of the things that's the most interesting to me that backs up some of Isabel's work that she just showed you is woody plants tend to have smaller diameter vessels, but then they have a lot of them. And when I think about woody plants, I often think of them as being water wasters, but this is a strategy that results in this sort of unique safety. You can still move a lot of water, but you're doing it such a way that your risk of cavitation and hydraulic failure is lower. And this is a pretty unique strategy and completely opposite of the grasses. So by using these types of microanatomical traits, you can draw more insight into the system. Okay, the last little bit that I have here with my remaining time is to talk about it, applying this on a landscape scale. So the headwater streams in grassy ecosystems like this one here have been declining in terms of their flow through the years. Where we work, we've been seeing declines for the last 30 years in headwater streams, and it was attributed to the increase of the riparian vegetation associated with these. So 10 years ago, we started an experiment where we clear-cut 3 kilometers of woody vegetation along these headwater streams to look at the increase in flow, and the woody plants were gone. And 10 years later, none of the water has ever come back. So there's no trees there anymore, but the headwater streams are no longer wandering. So Rachel, who works with me, did an investigation using stable isotopes. And so for the grass species, as you would expect, it was primarily using almost 80–90% of its water was coming from shallow soil moisture. The 2 trees, though, these are adjacent to the stream corridor, like within meters of the stream corridor, are using instead deep soil water. So water in this like 1 to 2.5 meter depth of the soil profile. And it was only this clonal shrub, the one that previously I said you basically can't kill, that's using undifferentiated. It's using all water that's available to it basically on the landscape. And it was the only one that was significantly using any of that stream water. So there's no direct linkage between these trees that exist on the riparian border and the flow of water in these headwater streams. So then we got to thinking, well, where is the water going? And one of the things that we've been postulating is that when you have a grassy ecosystem like this, because the rooting depth is shallower, you see basically potential infiltration pathways that look something like this. When you have woody plants across the topographically complex landscape, the roots go deeper and it forms these macropores or channels for water to infiltrate to deeper portions of the soil profile, such that even if you come in and you clear cut them, those roots are still there. And with those roots still being there, there's pathways for infiltration as they decompose. So water on the landscape potentially is not recharging that stream in the same way it once was, because you now have new channels for infiltration, basically allowing water to bypass that stream system. We wanted to scale this up. This is the watershed that we work on in 91 versus 2019. We used Google Earth like many of you have, and we basically went out and we quantified the change in shrub area on this watershed over time, going back nearly 30 years. And then we were able to, this is that watershed, we were able to take that data and using SapFlux data to estimate ET from these species, put some estimates on the amount of water that has been lost. So in 1978, it was nearly all grass. You can look at the flux of water both individually as well as across the whole watershed. But by 2020, it was nearly 80%. It was only 80% grass, so a decline of 20% and a 21% increase in shrubs. Granted, these shrubs are not filling bare ground. They're displacing grasses. And so this isn't a filling in of the system. It's a replacement in the system. But this process, by having the woody plants come in, results in a large increase in water use, such that a 20% increase in shrub abundance over the last 30 years has resulted in a 25% increase in evapotranspirative flux. So there's your water. This explains in this system where that water has went. It's through shrub expansion across the landscape that the streams no longer run, not the vegetation adjacent to the stream itself. So to wrap up, root distributions and densities can vary by these landscape drivers. Evaluating these root traits that I talked about can provide some key perspectives on bush encroaching species. And finally, when these woody species replace grasses, there's likely to be very large ET flux differences that exist across the landscape. Thank you very much.
Thanks, Jesse. That was a great talk. I have 2 minutes, so probably 2 questions. There's a question on the floor. You can put your hand up. Okay, Susan's at the back, and Barley will be there. In the meantime, does anybody else have a questions? We can probably just get the mic to you. Tasha?
Thank you. That was really fascinating. I'm intrigued by that xylem vessel diameter cavitation story in roots. I mean, I've thought about it in stems, and there's this relationship where the taller you get, the wider your xylem has to be at the bottom because it has to keep widening by a certain fraction to reduce the resistance. But I'd never really thought about the opposite, the mirror image below the ground. And so if you have really, really long roots that tap into groundwater, it will make you less vulnerable because you've actually got water. But at the same time, to get there, your roots have to be really long and your xylem presumably has to be wider, closer to the surface. And does that introduce potential vulnerability, especially before the roots are so long that they can reliably tap into a constant source of water?
Yeah, you're right. They have to be wider near the surface, and then they decline in aperture as you go down. One of the things that you will see in grasses, those deepest roots in the soil profile will just have one vessel, and then by the time it hits the surface, they'll have 5. Woody plants will start with only a few, and by the time they hit the surface, they'll have 20 vessels within that same vascular tissue. For deeply rooted species like phreatophytes that are using water from the water table, what you will see for them to accomplish that is they have to produce a lot of roots to get down there. So when you only find a few deep roots in a system, you have to think about what the size of those vessels are, because a few roots can only move a small amount of water. It's constrained by the size of the pipe. And so to move a lot of water, you have to have a lot of pipes. But you're right, the anatomy of building those is constrained from the bottom up to the top of the canopy and from the ground level to the bottom of the root tissue.
Thanks. Tasha?
Hey Jesse,
Hi,
thanks for
Tertia.
your talk. I just wanted to clarify, because you spoke about how that one reference you had in the groundwater journal was talking about how the roots behave and the preferential pathways in limestone geologies. I just wanted to find out your Kansas site, what kind of geology is that? Is that also limestone?
Oh, yeah. So we're a karst landscape, but we're kind of early in that karstic development. So we're mainly limestone and then what we would call mudstone, which is like a shale. It's a permeable shale. So we see water moving in our system in between these 2 layers of solid rock and more weatherable rock. So we see a lot of lateral movement, you know, in that case, between this limestone and mudstone. But we're very karstic. And so as those roots get down there and they produce carbon dioxide, it acidifies that soil and opens up new pathways for water movement.
Okay, cool, makes sense, thank you.
Thanks, Jess. Maybe just a question that I have. Your woody vegetation, or the big sort of riparian that came about on the stream, was that alien or was it indigenous riparian, so to say?
Yeah, no, our woody encroaching species is native. It's been there. Historically, it was confined only to these riparian corridors. So the question, but it's been now increasing in abundance for the last 30 to 40 years, where now if we don't burn every basically 2.5 years, we lose the ecosystem. And so, but it is a native species that is doing this. And a lot of the woody encroachment in the central Great Plains portion of the grassland region of North America is native species that are doing this. So it gets at the point, you know, why? Like what has changed that these species that were once kept in check are now have run amok?
Okay, thanks. So I was just thinking in South Africa, there's a big alien clearing program to sort of, you know, it's taking a lot of the water in the head, or the streams from the streams stop flowing because of alien veg.
Yeah.
And there's a big clearing program that's taking place. So I just thought, well, maybe there's some similarities there.
Yeah.
But thanks, Jesse. That's excellent. Okay, so I think we have quite a good audience this time around because we're moving off to a new theme, which is the freshwater theme and wetlands. So thanks for those who are staying. And because you are staying, Judith, can I probably hand over to you? We have some surprises under your seat.
Thanks, Robin. So we're having a quick commercial break. So I'd like everybody to please stand up and turn your chair over. If you find something stuck to the bottom of the chair, please bring it to me. And this isn't a catch. I'm not going to ask you a question or ask you to dance or sing tonight. So there should be 6. So have a look. They're quite clearly marked, so you'll easily see them. Okay, great stuff. We've got one. Come to the front, please. So if you have empty chairs next to you, you're welcome to turn those over too, please. So I'd like to get all 6. Pleasure. Yes, that's one. Come along. Thank you. Okay, we should have 2 more. Okay, last number. Well done. Thanks, everyone. So I'm going to hand back to Robin, and he can just tell you a bit about the book.
Okay, awesome, guys. So, actually, who has a book? Jason, can you just keep the book up? Sorry, I gave all the books out. So, there's a nice book there. It's called From Sea to Source 2.0. So, it's a book that I've been working on and a couple of colleagues here that we not worked on. I never wrote it, but I just co-authored a couple of chapters in that book. But the book is basically a global assessment or global case studies on restoring river connectivity in the world and providing barrier-free migration routes in a lot of river systems across the globe. So it's a very interesting book. It's case studies from all over the world. And yeah, enjoy the book, guys. Thanks. So just quickly moving on, we'll carry on with the program to keep on time. Sorry, where's my program? Over here. So Dr. Gordon O'Brien. Been working with Gordon for many years now. Thanks, Gordon. And you got a very long title, Gordon. I think you can tell them what that title is. Thanks.
Thanks, everyone. Thank you again for the opportunity to present. I'm going to shorten it. So what I'll present to you today is some of the work that we've been involved with. Is this backwards, forward, perfect, big buttons. I'm going to present some sort of highlights, outcomes of some work that we've been undertaking across Africa on the management of multiple quantity, quality, and other stresses, so multiple stresses affecting the social and ecological endpoints of vulnerable African water resources. I often start with this diagram to represent, this is a very simplistic representation of a socio-ecological system related to a river that we're interested in. And even from this, it just highlights how dynamic these systems are and how complicated, how many processes, how many components there are. And when we start to compare social and ecological systems and we look at them as a system holistically, how dynamic that can be. And this is the challenge that we face, is trying to represent these systems and trying to understand them, because this is what we care about. This is what we want to manage. But in a multiple stressor context, this is often what we are dealing with as well. So not only do we have these dynamic ecosystems with their own processes, but we have a plethora of multiple stresses affecting these ecosystems. And this is what we care about. We care about trying to manage these stresses in the context of these socio-ecological attributes of the systems we care about. The approach that we have established and been working on for many years now is ecological risk assessments. And quite simply, it looks at the probability of a hazard occurring or any stressor occurring in the environment, and then the exposure and ecological effect relationships. How does it enter the environment? How's the environment sensitive to or responds to that stressor? And if we can understand those relationships, we have an opportunity to estimate the risk of those stresses to these endpoints. We have a range of publications, so if you look it up, hopefully you'll find some of our work. But what's special about the regional scale ecological risk assessments, it's a progression from the traditional risk assessment approach where you're working with a stressor, a receptor, and a response with the exposure and ecological effects relationships that makes it a risk assessment, to regional scale risk assessments where the location of these different sources of stresses, the relationships between sources of stresses and stresses, the relationships between the stresses themselves and the receptors and multiple receptors within habitats, and then importantly ecological or ranked ecological impacts and how that affects the socio-ecological system that we care about. And if we understand where they occur in a regional context, we can start to apply this and attempt to manage these ecosystems on multiple regional scales. So I'm going to give you some quick feedback of case studies that we've been involved with across the whole of Africa. Wonderful opportunity to work on these dynamic ecosystems across the continent. And here's a quick methods just to show you how these projects usually unfold. So any given river ecosystem, we're generally looking at them on catchment scales, but we identify reaches of those rivers throughout the catchment that represent the variability of those river ecosystems, usually from the source to the coast or that regional area that we're interested in. Some of the wetlands that we've been working on in Africa are many hundreds of kilometers long and across. So, in those instances, we're actually only focusing on the river and the upstream catchment. So with many colleagues, we characterize the hydrology, water quality characteristics, and we've relied on and very importantly been working with some hydraulic assessments. Not only single cross sections, which they refer to as one-dimensional cross sections, hydraulic cross sections, to be able to represent the flow in ecosystem characteristics, but more complicated two-dimensional hydraulic where reaches of rivers have been modeled. That's been very useful for us so that we can look at and then try and understand the dynamics of different flows in the river channels themselves. And this is actually a very simple hydrodynamic model of the Crocodile River. What we do then is, with ecologists come together, and we're usually involved with a range of microbiologists, fisheries scientists. I usually am a part of that team. Vegetation and invertebrate scientists generally. We've also had mammalogists and other scientists, especially in the Sahel region. We've worked with birds, for example. And we come together to try to characterize those important processes that represent the ecosystems, not only biodiversity, life cycles of species, but important ecosystem processes. And that all culminates with an understanding of how the species, populations, and communities respond to driver variability. We're starting to pull relationships together between the flows and how the ecosystems response, as well as water quality, habitat characteristics, and the responses of ecosystems, and importantly as well, how these variables, environmental variables, affect ecosystem services. So once we've used this information to establish these nice response curves, and there's a plethora of tools that we can use to generate these relationships, we often use that information to give us these independent requirements of ecosystems. So in an environmental flow context, which is an overlap of the type of work that we're involved with, we at that stage start to provide these requirements of ecosystems, these sustainable development goal targets, and in a South African context, these resource quality objectives related to these drivers that are affecting the ecosystems. But we've taken a very big next step where we know that these indicators out of context of this dynamic integrated ecosystem is not adequate. So we use Bayesian network probability modeling tools to integrate and look at the cumulative response of ecosystems and the synergistic effect often of these multiple stresses affecting the ecosystems that we care about from a social and ecological context. And then with that information, we provide new requirements. We update the requirements that were provided, usually provided without the consideration of the synergistic integration. And then we're able to, in adaptive context, learn and test and improve our understanding of how these systems respond to these stresses. So that's what we do. It sounds so easy. This is some of the outcomes, and this is what I really wanted to focus on today with the rest of the time that I have, is to show you what we're doing with this information. So this is the Sud wetland, and what's really interesting is that we're starting to demonstrate, using a lot of these approaches, what really is driving these ecosystems. And often it's nothing close to what we imagine. And really interestingly, from a Sud and Central Africa. I'll get into more detail in a moment, but this is just an indication to show you what the relative risk outcomes look like. We're looking at the probability of occurrence of different scenarios. So you don't need to look at the details, just want you to see that we have comparisons and differences where we've selected different endpoints and we've looked at the different risk to ecological and social endpoints and how they compare with each other. And if you unpack that, what type of information do you have? So in North Africa, in the Sud and the Inner Niger Delta, I think I've got a slide on that as well, we're in a position where supply exceeds demand. So we've argued that in many parts of Africa, the 2030 SDG agenda will be achieved, but it's by default. It's just because we haven't developed those resources yet. That doesn't mean that the trends in change is changing considerably. And that's something I wanted to highlight as well here. We've applied this approach to the Lesotho Highlands Phase 2 project for the determination of environmental flows. And one of the interesting outcomes was to reduce the very high risk, the high portion where there's a 27% chance of failure of a fisheries, for example. We were able to mitigate that risk by proposing the establishment of fishways and improving fish connectivity in the basin. So it's quite dynamic and gives us multiple opportunities to look at trade-offs and other opportunities to mitigate risk before a lot of these developments occur. This is an example from the Tugela River catchment in KwaZulu-Natal, where our preliminary risk assessment kind of indicated that there wasn't a lot of going on. When we implemented it and tested it, we found that our estimates weren't accurate and that it was a lot worse than we initially predicted. We also improved our confidence to demonstrate that one of the Mandini streams, for example, was particularly problematic. We then used the approach to demonstrate what would happen and where we were going if we just continued with business as usual, but what it would take for us to achieve this sustainable balance between the use and protection of these resources. And unfortunately, it ends there. We wish that more of this would be taken up. So we also are able to unpack it and demonstrate what is contributing to risk, because in these case studies, everybody's pointing fingers at each other, and we're also able to identify the relative importance of individual ecosystems and how vulnerable they are to changes and these multiple stresses relative to each other in these spatial scales. This is some of the really initial work that I did in the around 2008, 2010. And I just wanted to show here that in the Vaal River system, looking at a risk assessment to yellowfish in particular, we actually forecasted how the system would change and where it would change and how vulnerable the yellowfish community was. And during a lot of the problems that we've experienced with wastewater treatment works over the last few years, we identified that then and actually gave recommendations to the Department of Water and Sanitation, and we just politely reminded them that we had predicted this beforehand. So it's very useful, and again, we just wish that a lot of this would have better uptake. This is a case study in the Mvoti catchment where we look at the relationship with, and what's important here is we can see what the downstream influences are of upstream land use developments. This is the Mvoti catchment in KwaZulu-Natal with industries, a lot of dams, sugarcane and other agricultural activities, and then the overall risk profiles. Work in the Mara River, again, what was really interesting here was with the Mara at the base here, Mara and the Serengeti are down here, and really importantly, what we wanted to identify was that the plans that they had to develop the resource, what the implications were, and we were able to divert some of those high impact development activities to work towards a better sustainable balance and environmental flows as a tool to implement those requirements. So it's been very useful. This is the Inner Niger Delta in North Africa, and just to show it's really special to work in these case studies where there is very little risk associated with developments because these systems are just so massive, and it's quite exciting. So that's wonderful context to us. But, and just to end with, I wanted to present some results from a recent environmental flow relative risk assessment study for the Limpopo catchment. This is where the wheels come off. And unfortunately, the Limpopo catchment, everybody knows, is one of those shared transboundary resources where it's everybody's basin. So in effect, it's actually nobody's basin. Nobody actually needs to take responsibility for it. And these are some of the risk assessment outcomes to look at the overall response of multiple stresses. So from our model, what's really important is that we we've got some indication of what it looked like historically, so we use historical information, and the model should represent something that we can justify, that it is responding correctly to available data and available scenarios. So based on flows and other synergistic effects with the early 1900 scenario, that's what the risk to the fish communities were in the Limpopo Basin, and that's where we are today. You can see a massive shift towards the threshold of potential concern with the Olifants River catchment, standing out like a sore thumb as usual. And then with the environmental flow assessment, now the red limit is unsustainable and beyond the limit of sustainability. So with the environmental flow requirements, we have identified and proposed requirements to maintain the system in a hard working state. But we've actually discovered that the evidence that was available to previous studies and our evaluation of that evidence with an improved understanding of the system and how it interacts suggests that the environmental flow requirements that have been gazetted are actually not sufficient. So that's been some interesting outcomes. And then if you prolong the worst 5-year drought into a climate change scenario, it's doom and destruction. So some quick examples of fish and inverts looks a lot better. The environmental flow requirements are a lot better. And then if you look at droughts, some really interesting dynamics with the upper Morocco Basin, and then riparian vegetation, if you look at that as a component and how there's massive changes. But the modeling is working very well. But what's really important is, and this is where we just use the different colors to highlight the range of variability so that you can identify across the basin where are the different stresses relative to one another. What are those areas that are hotspot areas? This allows us to consider trade-offs and where to direct our management efforts. And on the top row, I've again just presented fish, invertebrates, and riparian vegetation, but at the bottom here, I've presented subsistence fisheries, water disease vectors, and vegetation for subsistence. And that was really interesting. So when we started to incorporate the ecosystem service considerations and where vulnerable communities are, and what the risk of multiple stresses are and how that will potentially impact vulnerable human communities, it again influences the way that we are starting to allocate resources. And very importantly, we have the ability now to demonstrate how these communities and how there's been such a shift from historical conditions and changes or increases in risk associated with multiple stresses, not only affecting the ecosystems and their functioning, but also other ecosystem services. So we hope this information will contribute not only to updating and improving our targets for sustainable development as well as resource quality objectives, but also to help us to implement a lot of these objectives. Yeah, that's it. And then just last slide to show that it's actually contributing to policies and things, which has been very useful. That's it. Thank you.
Thank you, Gordon. Thanks for the really nice talk. I'll squeeze in one question. If anybody has a question on the floor. Nope. Okay. Thanks so much, Gordon. Okay, so Stephen Hoodborn is not available to give his talk, but Albert Meiberg is going to give his talk. Albert? Thanks, Gordon. Okay, cool. Let's come up. And so Albert will present to us on characterizing the algal blooms we experienced in 2021 along the Massingier Dam. Thanks, Albert. You can go ahead.
Thanks, Rabia, and thanks, Gordon, for setting the scene so nicely. I think that big red area right at the end of that in the Olifants River is the Olifants River Gorge, and that is where this work has been focused on for the last 12 or so years. So yeah, as I said, that's the Olifants River Gorge. That's where it is. It starts at the confluence between the Olifants and the Letaba rivers, and it ends at the mouth of Lake Masenga in Mozambique. So half of it is in South Africa within the park, and then the other half of it is in Mozambique, just above Lake Masenga. This was the site of a mass mortality of crocodiles between 2008 and 2010. About 200 crocodiles died, and they died from a disease called pansteatitis. It's a dietary disease, and it's associated with the consumption of polyunsaturated fatty acids and vitamin E levels, and it is caused by the inflammation and necrosis of adipose fatty tissues, which renders the animal immobile and incapable of utilizing its fat reserves, so the animal normally dies of starvation or drowning. David Hoogsemeyer's work in 2010 to 2012 identified that pansteatitis was not only occurring in crocodiles, but also in catfish within the same area, and with a similar etiology to that in crocodiles. So the probability of this causality being different in the same location was quite very low, and they attributed to the same mechanism. And so catfish became a really good proxy for detecting pansteatitis within the system. Just a quick overview for those who are unfamiliar with stable isotope analysis. In aquatic systems, we use stable isotopes of carbon and nitrogen to define the relationship between animals in a system, where you normally get your primary producers on your bottom left and your top predators on the top right corner. And if we look at some early stable isotope work that was done in the Kruger Park during the initial pansteatitis outbreaks, we can see that. Let me just explain quickly. Sorry, so you're looking at nitrogen there on your y-axis and carbon there on your x-axis. And this is the system that was suffering from pansteatitis, so here called the confluence. And if we look at a normal system like the Crocodile River, the green represents your invertebrates, and the blue and red represents tigerfish and catfish within the system. So we would expect tigerfish to have a higher trophic level as they're obligate piscivores, and barbel, being omnivorous, will have a varying trophic level. So in all of these systems, it's relatively normal. I mean, we get invertebrates at the bottom, and then following up from all of those, we get the catfish and the tigerfish. But in this system, there was a relatively big gap between the invertebrates and the fish, and so there was something missing in there. Follow-up analyses of that attributed that missing gap to filter-feeding fish, and the initial speculation was that it was silver carp, as they were quite prolific in Lake Masinga, and it was thought that they were moving up into the Olifants River Gorge and filling that void niche. But after the 2012 work, I think in 2013-14, there was some initial analyses on fish from Lake Masinga, and there they found that it was actually Mozambique tilapia that were filling that niche. And Mozambique tilapia is a fish that's quite prolific throughout all of the systems in the park, and it was filling that niche pretty well here. But what about the crocodiles? If we plotted the crocodiles in the Kruger, we can see that the niche we would expect crocodile diet to be does not overlap with fish. Crocodiles were actually plotting below fish, so if you interpret this at face value, it would look like fish are eating crocodiles, which is obviously not the case. And this open niche was either filled by a terrestrial dietary source or it was an aquatic macrophyte, and the probability of crocodiles eating plants, I think, is very low. And so we did some follow-up work throughout the whole Olifants River system, and we could prove that crocodiles actually don't track the nitrogen profile of fish within the system, and they're actually more dependent on a terrestrial dietary source. So the main dietary source for crocodiles in the Olifants River system is not fish, it's terrestrial. This has been published in both Woodbourne, and then I've got a paper currently in review on this. And we could use the crocodile diet-to-tissue discrimination factors to predict the diet where we would expect it to be, and it's definitely not fish. But that doesn't mean that crocodiles never eat fish. If you look at these time series analyses, I don't know if you can see the little serrations on the crocodile claw there. We use these to create time series dietary constructions for crocodiles that succumb to pansteatitis. And if you look at those, we can look at the growth rate, and we've done some radiocarbon analyses to look at the age of these claws. And we've discovered that in some of these crocodiles, we see these annual spikes in nitrogen values, which we think are episodic fish binge feeding events, which were prior to the pansteatitis outbreak. So some of these crocodiles, this is when they died at the end on the right-hand side there. Just before their death, we see this upward trend in nitrogen profile, suggesting that they were possibly moving towards some food source that had elevated nitrogen values. Now, if you work in the stable isotope world, you know that pollution in aquatic systems is normally associated with relatively high nitrogen values. And this became known as the filter-feeding hypothesis. This was hypothesized in 2012, and we've since done a few studies to look at the mechanism. But just briefly, Lake Masinga the dam wall of Lake Masinga was raised in 2007, and this causes a backflooding of the lake into the Olifants River Gorge. So normally where you would have pollution from the Olifants River, which is polluted all year round, it's one of the most polluted systems in the country, flow into the lake and get dispersed throughout the whole lake. It was now accumulating within this narrow gorge area, which had changed from a river system to a dam system. And this causes a prolific algal bloom, which then attracts a lot of these filter-feeding fish, which are readily available for crocodiles to consume. And so there's a pathway or a trophic linkage between the algal blooms and the crocodiles, and that's the fish that congregate in this area. Just as a little illustration, this is before and after the dam wall was raised, and that little red area is the same area in both images, and you can just see the inundation of the gorge after the raising of the lake in 2007. Just another illustration, this is on the border between South Africa and Mozambique, and that red arrow is the same red arrow in this one. So that's the difference between a river system and a dam ecosystem caused by the raising of Lake Mzingi's dam wall. But then the question was, is this a one-time event? Are we going to see pansteatitis again? And what are the conditions that are needed for this thing to reoccur? And after 2012, we've done a lot of analyses, but we've never had the conditions that we've had in 2012. So we've never had the high lake levels and the high flow levels because we had the drought in between. And so we were focusing on river processes. I'm not going to go into too much detail here, but basically the Olifants River in the park follows what you would expect from a maturing river. Food webs in lower reaches rely on these allochthonous nutrients, not autochthonous nutrients, so nutrients that are produced elsewhere and transported to that site. And along the river, we see a breakdown of organic material during low flow, and these are incorporated into longitudinal food webs and mediated so that the Olifants River in the park, which is a relatively natural system, serves as a buffer for pollution outside the park. This graph is quite complicated, just bear with me. So on this axis, we have the lake level of Lake Mzinga. On the other y-axis there, we have the river flow, which is represented by the red. So the lake level is blue, river flow is red. And then we used satellite imagery to look at when the system was flooding and when there was an algal bloom in the system. And so all of the dots that you see in orange is a flooding event, and I'll show you some satellite images now. And then those in green are algal bloom events, either within the dam or in the gorge, depending on where the lake level is. The red line that you see here is the level that the lake needs to be at to backflood into the gorge. So when you have an algal bloom and it's below that red line, it's not an algal bloom in the gorge, but a dispersed algal bloom within the lake. And we've never had the conditions that we've had in 2008, except last year in 2021 when we had a lot of rain in the system, there was a lot of flooding coming down, and the dam filled up relatively quickly. So just to illustrate that, that's before the rain started in 2020. This is the first floods that came down, filled up the dam relatively quickly, and you can see these sediment-laden waters. Now, what's interesting about these waters is that it's bringing with it a lot of nutrients, but because it's so sediment-laden, there's no penetration of sunlight, so there's no algal production, there's no remediation. So all of these pollutants are just flushing down the system and getting dumped in the dam. So as soon as there's any form of sunlight available, I'm sure you can start to see on the right-hand side there, we've got algal bloom starting in the gorge. This becomes quite a bit more pronounced as the system slows down and these floods stop, and we see the algal bloom starting to accumulate more. We still see some sediment in the system. But what happens when the system slows down completely and you have this low-tech system that's not flooding anymore, so there's no more sediment transport coming down, is there's a prolific algal bloom that goes and covers the entire dam. If you just zoom up, you can see that that algal bloom front moves up the gorge as that area becomes less and less turbid and sunlight can penetrate the system. We took a helicopter and flew there just to make sure that the satellites aren't lying, and this is what it looks like. Extremely green, soupy water with microcystis confirmed in them, so a prolific algal bloom in that system. There's some water quality analysis just showing that it was off the charts, hypereutrophic, just as we predicted, but there was no pansteatitis. So what's the missing link in the system? Because the mechanism suggests that when this happens, there should be pansteatitis. This is just an image to show the entrance of or the mouth of Lake Mazinga. So on the left, you can see the relatively narrow mouth, and it opens up into the massive lake here. And then if you turn 180 degrees, you can look up the gorge. And if you're a fisherman, that's a pretty good place to put your net, instead of somewhere in the open water there. And so when we went there, that's exactly what we found. Buckets and buckets full of Mozambique tilapia and hundreds of meters of nets constraining the movement of fish up into this gorge. And when we look at crocodiles congregating close to the mouth of Lake Mazinga, of this, I think there's 60 odd crocodiles in this, 3 of them had nets around their mouth. So they were actually going for these fish, but the fishermen were getting them before them. If you look at satellite images between 2005 and 2021, we could see quite prolific expanded fisheries around this area, and we know that they export a lot of these fish to Maputo. So in summary, pancreatitis has been linked to filter feeding fish and nutrient pollution in the Olifants River Gorge. It requires a large population of filter feeding fish in order to get a crocodile sick, basically, and to form a trophic link. And currently, overfishing may be responsible for reduced fish populations, and the top aquatic predator in this system looks like it's changed from crocodiles to people. And this is quite frightening because people are utilizing fish that we know are exposed to microcystins and toxins and exported pollution from South Africa. It's also bringing people into quite a human-wildlife conflict situation where they have to deal with crocodiles on a daily basis. Thank you.
Thank you, Albert. That was a very good talk, man. Thanks. I have 3 minutes. Right, Sam Ferreira has a question. If you have a question, put up your hand. We can try to get the mic to you so long.
Albert, thanks for that. A nice summary. Some of the work I didn't even realize is telling us this nice story. But I was just wondering, I think it's really an interesting consideration about the high filter feeder requirement. But there was a management intervention that we did as we were building up, and we saw this algae bloomers coming, which was really actually providing an alternative food item for crocs. Have you got any sort of ideas whether that would have had an effect?
I don't know because we didn't quantify it, but I'm sure that it had. I mean, we went there during that prolific algal bloom and we tried catching fish and we just absolutely got nothing. We had seine nets dragged through, cast nets, rod and reel. There were no fish. I think we caught 1 tiger fish in 2 days of sampling. So I think definitely there was an alternative food source that's available in the absence of the fish, but it may be good that the fish are not there because, I mean, that's the trophic link to these crocodiles.
Thanks, Albert. There's a question from Danny.
Thanks, Albert. I think, pancreatitis and dietary diseases are sort of 2 factors. The one is that you consume a whole lot of stuff, and the other is that you lack the enzymes or dietary mechanisms to deal with it. So when you're eating a lot of polyunsaturated fatty acids and you've got a lot of antioxidants, it's fine. If you don't have a lot of antioxidants and you live in a system that's polluted, for instance, it might be problems for you. With that in mind, do you think that humans are genuinely at risk?
I don't know. To be honest with you, I've no idea. I don't know what the mechanism is in humans. I don't know what the physiology is. So I think that's a very good topic for a future study. And actually to look at the levels of fish intake, because I think that a lot of these fishermen in Lake Mazinga are actually catching these bucket loads of fish, but they're selling it. They're not eating most of it. And so to look at where these things go and how they get incorporated into the anthropogenic food web, if you can put it like that, is quite an interesting study.
Thanks, Albert. One last question.
Great. Thanks for a great talk. I think we're expecting another big die-off with the Lawscope Dam coming this way, I think. Just, has anyone done surveys of fish in the olifants? Maybe there's just no fish, the crocodiles, because of all the pollution from coming through from Bootbank and all that.
Yeah, there's actually been quite a lot of studies on the Olifants River system and the different fish. I've been involved in campaigns where we've caught hundreds of fish. I mean, you throw a cast net and the whole thing can't even pick it up because it's full of Mozambique tilapia. So there's definitely fish there, yes. I think it's just the overfishing pressure that's currently coming in from the Mozambican side that's maybe limiting it in that region where we see the algal blooms.
Cool, thanks Albert. Thanks man. So this is the last talk for today. Stephen Koza unfortunately couldn't make it, but Peter Botha, who I think was a co-worker on the project, will take us through. Thanks Peter.
Good afternoon, everyone. Peter Botha. I'm here to present some work that a colleague of mine has done, Stephen Koza in Marakele. And it looks specifically into the hydrogeomorphological characteristics of two sloping mares in the Waterberg Mountains. For those of you, just as a bit of background with regards to mares, bogs, fens, or peatlands as they are commonly known. They are normally deep accumulations of incompletely decomposed organic material. Carbon sequestration that took place deposits that material within the peatland. The biomass exceeds the long-term loss because the breakdown or decomposition does not occur completely, and the hydrological flow within an atmospheric decomposition is limited. 3 to 4% of the world's surface area are peatlands, and that 3 to 4% represents 30% of our terrestrial soil carbon available to us. The areas are normally reasonably species poor, and it's a very unique type of setup within a peatland compared to what is happening upstream or outside on the boundaries of the peatland. All mires are wetlands, but not all wetlands are mires or peatlands. And then a question that is recently been asked is what starts happening when we've got peatlands that rise out with the decomposition starting to take place, and especially the methane production occurring from that decomposition that's taking place. Within Limpopo province, there are several mires in the Waterberg region or associated within the Waterberg. The Matlabas Mire in Marakele, just north of Tabazimbi, Pitse, Bakovenkrans. That mire caught fire 2 years ago, and we are currently in the process of trying to curtail that fire that has burnt a big portion of the peatland there. Wonderkrater, which a lot of you might know, and then Nailsoog itself, just north of the town of Warmbad or Bella Bella. The Matlabas Mire is situated within the Waterberg Massif. It is the headwaters of the Matlabas River, which is a huge tributary of the Limpopo, draining northwards from Marakele onto the low-veld areas of Limpopo, reaching the Limpopo River. If we look at the two specific mires that's in question here, both the one in Nailsoog, which is a private reserve, and the one within Marakele, both of them were used extensively for agricultural production and use over the years. Marakele itself was farmland, being farmed right into the 80s, early 90s, and several impacts have occurred along the way, from soil erosion, overgrazing, animal feeding points, watering points, to invasives and the planting of certain species that should not be occurring in these environments. Just as a matter of interest, that mire picture on the left there, that's the impact of large herbivores on peat domes. The Marakele mire, several transects were placed across the mire, trying to understand the interaction between surface runoff and our groundwater regime, what is happening within there, because it's important for us to understand the balance and what is the driving force behind the development of these mires. We use the process of installing piezometers with wells, and we refer to these as nests, to try and understand where the source of the water is and what the pressure is that is applied by the artisanal water coming out from below these mires. if we look at the Marakele Maya, or the Matlabas one, the expectation was that there's surface flow involved, there's intermediate flows, seeps, and then artisanal water that is provided from below the surface, below the peat deposits. And what was quite interesting from the information that was gathered, the data, was that there is a balance currently in play within that system where there's upward pressure from the artisanal source counteracting the erosivity from any surface flows occurring within the peatland itself. If we look at the Nyloog, or Nyloog landgoed as it's referred to, in the Waterberg, which is about 150 kilometers southeast of the Matlabas, several impacts was listed there during the process of setting up rehabilitation plans in there, all related to agricultural use and historical use within the landscape. This specific farm was one of the first farms also provided or mapped out as private landowners within the Limpopo area. It is a farm that has got a long history to it as well. The peatland within this system is quite unique and pretty small. It is only about a hectare in size that the peatland is situated in the Nyl Rivier, which is the headwaters of the Nyl Rivier feeding into Nyls Vlei, which is an important Ramsar site for us as well. The groundwater monitoring, as I said or alluded earlier on, we used piezometers and nests that was installed, and the same was replicated here within this peatland here, with transects placed strategically across the peatland there to determine the water balance within the peatland. The geomorphology that came about from sampling within this peatland here, where we realized we've got a clay layer situated below it with a peat layer on top, accumulating on top of the clay. The hydrology of the Maya is also within the same balance and the same characteristic as the one in the Matlabas, where you've got dispersed surface flow coming from the surrounding catchment, channel forming at the bottom, which is similar to what happened in the Matlabas at the exit point of the Maya there. We've also had channels occurring, eating back into the wetland there, or the peatland, and ground water flows or artisanal water flows within that. The same setup here as well, as I said, where the erosion is cancelled out per se by the upward pressure from the artisanal springs. The upward pressure from that maintains the vegetative growth and development within the peatland, creating a surface roughness that counteracts the action of the surface runoff from these areas and providing resilience to this landscape, preventing it from scouring any further and any further channels forming within. The equilibrium results prevent further channel erosion, stabilize disturbed environment, prevent any potential head cuts and erosion, prevent further sediment deposition downstream, and create an environment for indigenous vegetation to establish. So it is important for us to note that should the groundwater source be impacted upon, the chances are that we will have a loss of some of these peatlands. Peat, once it's dried out, become hydrophobic and it cannot be re-wetted. It enters the system, decomposing, and then we also have to take consideration of the methane produced from these peatlands. We followed an approach in both of these where we opted to go what we call a soft approach in trying to curb the potential impacts, especially from the gully erosion starting at the exit points of these peatlands. And we've opted for the softer options, not to introduce any real foreign materials into there, and with a slight bit of help getting the peatland to restore itself. And it was basically to prevent erosion and trap the sediment coming down there. Low flow energy areas was targeted. Rock and soil blankets were utilized, and I have to emphasize with rock the least viable option that we try to steer away from. Earthworks sloping infilling, and especially with the product that's seen there in that bottom picture called Magmat R, had huge success. The moment these were put in situ, we had an immediate vegetation response, and once the vegetation got going, we had an end to the problem of the channeling within the wetland. Thank you.
Thanks, Peter. I open up the floor for 1 or 2 questions. Eddie at the back there.
Thanks very much, Peter, for that presentation. I've got a question around that region, the Western Limpopo, the western portion of the basin. If you look at the climate change forecast for that region, it basically shows that there might be winners from a hydrological perspective in terms of facilitating groundwater recharge, but then obviously you're going to get drought stresses as well. So my question is, in terms of maintaining the ecosystem services from those headwater mires in terms of carbon sequestration and groundwater baseflow, what would be your hunch in terms of where to prioritize action? You know, in terms of, is it managing an erosion problem, or is it managing some other aspect that you have to maintain in that ecosystem? Thanks, Peter.
Well, it's a two-pronged approach. So making sure that what is there remains there at this stage is critically important, because twofold. Once we lose it, we cannot get it back. Peat is not something that is formed overnight. We're literally talking thousands of years, millennia, for the accumulation of the peat. That is the one thing. So we need to keep the peat in situ and prevent that methane from going into the atmosphere. The other thing is, from a regulatory perspective, it is critically important that we start getting a grasp as to what's happening with groundwater withdrawals within our landscape. It is a challenge for everyone, everywhere. That there, at the moment, is a huge drive from the agricultural sector, and potentially even from a social drive at the moment as well. If we look at municipal services collapse with regards to water quality that is no longer be usable, they simply tap to the easy source, and it's the groundwater source. One prime example at this stage is Mafeking at this stage, that is just now started considering pumping water out of mires around them as well there in Northwest. So yes, it's a two-pronged approach that definitely has to be looked at.
Thanks, Peter. Thanks for that. Okay, guys, that brings us to the end of today. I think just the last final announcement that Samantha will be here at 7:15 tomorrow morning. So the guys who are presenting for the opening session, you can load up your talks and just get an idea of how the system works. Judith, is there anything else you'd like me to mention? Is dinner at 7 o'clock? Okay. So dinner is at 7 o'clock this evening at the golf club. Please bring your ticket along. If you don't have your ticket with you, you will not get a plate of food. Okay, anyway, guys, enjoy the evening, and thanks for the great day.